Hi, my name's Richard. Welcome to another one of TMAG's live Facebook tours. It's the beginning of National Reconciliation Week, and TMAG would like to acknowledge and pay respect to the Tasmanian Aboriginal community, the First Nations community of Lutruwita, Tasmania, the island that we're on, as the traditional owners and continued custodians of the land. TMAG is on Muwinina land. Now, because of the history of what happened here, there are no Muwinina people or their descendants left. But the First Nations community, the Tasmanian Aboriginal community, is still very strong in this place. And I'd like to acknowledge the elders past and present and those emerging as well. Now, we're at TMAG, but we're at the Tasmanian Herbarium, which is on the Sandy Bay campus of the University of Tasmania. And the herbarium's part of TMAG. To meet a special guest presenter today, so let's go inside. Hi, Miguel.
Welcome to the Tasmanian Herbarium. This is a herbarium is a term that we use to mean two different things, either a plant collection or, in our case, the place where we store this plant collection. So let me show you around.
So this is Miguel de Salas. He's the Senior Curator of Botany here at the Herbarium.
That's right.
We're just going to go up and have a look up the corridor.
We're going to go into our main vault, which is the place where we store the main plant collection, if you want to follow me. If you follow me under here, now we're just walking underground. This is the main vault, storage vault at the Tasmanian Herbarium. Now, the Tasmanian Herbarium is the Tasmanian Museum and Art Gallery's collection of plant specimens, botanical specimens, which is also the place where we carry out research on those plants for a lot of different purposes. We're here in an underground vault because being underground keeps the temperature and the humidity steady year round, and it's a really good place to store delicate objects like plants without them deteriorating. So you can see we have a bunch of compactus units and boxes, and each box is full of herbarium specimens, which is plants attached to pieces of card and classified. And so I'm going to be talking to you a little bit about the system that we follow and how that system started and the sort of research that we do. If you'd like to follow me, I've set out a few specimens in our preparation room.
Miguel, how many plant specimens would there be in the herbarium altogether?
Our collection, we don't know exactly, but the Tasmanian Museum and the herbarium collection contains somewhere between 250,000 and about 300,000 specimens of plants, but also algae and fungi and so on. And they're all stored in here. Most of those plants are collected in Tasmania. We have the largest collection of plant specimens from Tasmania anywhere in the world. But we also have specimens from various other parts of the world. A lot we use for reference. For example, if you find a new weed and you don't have specimens from other parts of the world to compare it to, you might never be able to identify it properly. All right, follow me. If you come in here, this is our main preparation room. This is the room where we turn plants that come in into a herbarium specimen. Now, I mentioned briefly as you came in that a herbarium can mean both a plant collection and a building. And people have been putting together plant collections, let's call them herbaria, which is the plural of herbarium, for 300-400 years. And one of the things that people could do once you have a dried plant specimen is they could start to study them. And I showed you in our vault that a lot of our specimens are arranged by name. And where do those names come from? A lot of the research that we do here is called taxonomy, which is the study of species, the study of plants and animals and where one species ends and another one starts and how to classify them. So the first person who ever started all of that was a chap called Linnaeus, who you may have heard about. He was a Swedish fellow who worked mostly at Uppsala University. I've got a picture of Linnaeus in here with his best Sunday garb when he was a young man. And Linnaeus created quite a revolution back in his time. He lived in the early to mid 1700s, and his parents were naturalists, and he was a naturalist. He was very interested in the natural world, in plants and animals particularly, but he was, I guess we'd call him today, a bit of a neat freak. He had a big collection of objects, but he liked them all classified properly, and he just wasn't happy with the way that things were being classified back then. So if you go back in time to Linnaeus's time, if you wanted to refer to this plant here, which is what we call a Cape gooseberry, there wasn't a simple name in scientific literature like we have for it today. In fact, the name would have been a description of the plant, and in this case, being in Latin, because that's the language that they used for all scientific communications, it might be Physalis annua ramosissima ramis angulosis glabris folies dentato serratis, which means with bladders, annual, freely branched, branches angular, hairless, and leaves with a serrated edge. So this gets pretty complicated, pretty unwieldy quite quickly, because anytime you want to refer to a different plant, you've got to describe it. And Linnaeus just didn't like that. And so what he came up with was a system of classifying everything by breaking it into smaller and smaller groups, and so that you had little final groups of things that were closely related, which he called genera, and each one had a bunch of species. And he came up with the idea of just using the two names of the genus and the species to name them. So in this case, it went from that long unwieldy Latin description that I gave you to just calling Physalis angulata. So he came up with this system, not just of naming them, but also of how to classify them. And by breaking it into smaller and smaller groups, the largest ones being the kingdoms, for example, animals, plants, right down. And so he was using, this is the book that he published called The System of Nature. By breaking all living beings into smaller and smaller groups, you ended up with few enough species in each group at the end that you could get a handle on them and identify them. And so he wasn't just classifying. By classifying everything, he developed a system where you could identify them, and that was his big breakthrough, because before Linnaeus came along, if you wanted to know what a random plant was in the forest, the only way you could is if somebody had told you what it was. You had to get that knowledge directly from another person. Linnaeus invented this system of breaking everything down and classifying it, so that by following his system and reading the book, you could end up putting a name to just about any random plant you picked up. For the first time, you could just pick up a book, pick up a random plant, and put a name to that plant and know what it is. So that was a massive revolution in the 1700s.
So Miguel, every plant and animal today has that dual name then?
That's right. So that system he invented is the system that we use today to name all plants and animals and bacteria. There are other conventions when it comes to microscopes, but for all plants and animals, that's the system that we still follow today. And we do quite a lot of work, and I'll go into that a little bit here at the herbarium on how to keep fitting things into the system so that we can identify them. And that's a lot of that taxonomic research that I'm talking about. So Linnaeus had a bunch of students he called his apostles, and he encouraged these students to go right around the world and collect plants and animals and just try and fit more and more things into his system with the ambition of describing everything, essentially, which we still continue today. And which means once you've described it and fitted it into the system and put a name to it, you can identify them if you come across it again in the future. One of Linnaeus' students was this fellow with a beautiful hairdo called Daniel Solander, and he's here in this portrait with Sir Joseph Banks. You may have heard, or the names may sound familiar. Banks and Solander visited Australia with Captain Cook on Captain Cook's first voyage. They were the designated botanists on board, and in fact one of our best known plants, the Banksia, is named after Sir Joseph Banks. In these voyages they collected a bunch of specimens, not just around Australia but also the South Pacific, and some of the earliest specimens that we have in our collection here at the Tasmanian Herbarium were actually plant specimens collected by Banks and Solander, such as this beautiful fern from New Zealand, collected in 1769, so this year it's 251 years old.
Wow.
And this is a herbarium specimen. It's a plant that has been pressed flat, in our case attached to a sheet of card so that it's relatively safe to handle, and once you do that, the plant won't break anymore when you put other things on top of it, and you can keep it essentially forever. There were quite a few voyages of exploration once that scientific revolution got going, and in biology with Linnaeus played a big part in spurring on that drive to go to the corners of the world and find and classify all the plants and animals. Botanists came through to Australia and they were classifying and collecting plants all over the place. One of the most important to visit us was Robert Brown, who was the foremost botanist in the UK at the time, and he came to Hobart. He came to Australia with Matthew Flinders in the voyage of the Investigator, and he spent a couple of years around Australia, and in one of his many visits he collected this specimen in Hobart. And you may see, if I can point with my finger in there, it says isotype. This is a very special type of specimen. This is a type specimen, and a type specimen is, in science, it is the official reference specimen for that name. So this is pineapple grass, we call it Astelia alpina, and the reference specimen, the official reference specimen for pineapple grass is this. So the name is tied to this specimen.
So when someone else collects a specimen of pineapple grass, they could compare it to this to see whether it was
That's right.
bigger, smaller, hairier.
Yes, say you've got a weird looking pineapple grass and you're trying to work out whether it's maybe a different species. What you compare it to, the most important specimen you compare it to is the official reference, which is this specimen. Mm-hmm. Now, interestingly, the type of humans, Homo sapiens, is Linnaeus' own remains. And
so he put himself into the system.
I don't know if he put himself into the system or if somebody else did after he died, because you're not allowed to name things after yourself. Not that we are named after Linnaeus, but people can name things after you. But anyway, going on and on, Linnaeus started it all, but we continue it to today. We're still doing a lot of that taxonomic research here in the herbarium, trying to find new plant species. And among these, for example, I've got some violets that we've been working on recently. So this is a sweet violet that you might have in your garden. It's quite a common garden plant. You'll see here the scientific name is Viola odorata L. Now L. stands for Linnaeus because he named it in his very first publication on plants. And that's the name that we still use today. Incidentally, Linnaeus is the only authority who has described a plant who is abbreviated to a single letter, because he started it all. Mm-hmm. Now, we've been doing a fair bit of field work around Tasmania, and a few years ago on the west coast we found a funny looking violet, which I've got in here. And you can see there's a funny, quite unwieldy name in there called Viola Spur, Harmon River, blah, blah, blah. That's called a phrase name that we use as a temporary name for something we think is new, but we haven't got a name for yet. Eventually, we've done the research. I've compared it to all the other existing violet species in Australia, and I worked out that yes, it is definitely a brand new species, and so we named it Viola serpentinicola, or the serpentine violet. So there's still finding a new vascular plant, so a new flowering plant like this, nice and showy, is a pretty rare event these days in populated places like Tasmania. Not hugely rare, but unusual. But you don't even sometimes have to wander that far. You don't even have to go to the west coast. Just 2 weeks ago, I was doing some taxonomic research for our Flora, which is the publication in which we are trying to describe all of the different plants and flowers in Tasmania. And I was consulting all of the collections in the vault of this particular group called Pseudanthus, of which we thought we had one species. And it turns out there was a second species hiding among the herbarium collections, which is called Pseudanthus divaricatissimus, or the red flower Pseudanthus, which we had no idea was in Tasmania. And that didn't take a trip to the west coast, or in this case to Freycinet, where the specimen originally came from. This was already in our collection. It took 17 steps to walk from my office to the vault and pull the specimen out, have a look at it, and realize this was a new species we didn't even know we had. So we still got some pretty fresh specimens, pretty fresh discoveries coming out.
Yeah, that's a great story. Miguel, can you talk a little bit about why is it so important that we can identify plants? Why do we need to know exactly what they are or where they're from?
Even just from a practical point of view, if we don't have a name for something, it's very difficult to refer to it. And so you can even go to very practical needs, like some plants could be poisonous, or the ones are useful to us, or the ones might be in need of conservation because they're very rare. Now, how are you going to know what you should eat and what you shouldn't eat unless you know what they are? And if you don't have a name for them, it's very hard to know what they are. And similarly for plant conservation, for example, a lot of the effort to conserve plants, or with declared weeds or things like that, is based on legislation. And the law says that this plant is protected, or that this plant is a declared weed. You can't have a law that protects a plant or makes another one a weed or a prohibited plant unless you have a name for it to put in the law that says this plant is protected or whatever. So we can't really talk about things unless we know how to refer to them. It's almost as if they didn't exist.
Have you got some other things that you'd like to show us?
I did. I'd like to, if we can come around this way, I wanted to show you a few specimens that I collected earlier today. And here's a little beautiful $2 store plastic container, but in technical jargon we call this a vasculum, which is essentially a container where you put damp paper to keep plant specimens fresh until we can deal with them back in here. So I've collected, here's a silver wattle, I have a grevillea, that's an ornamental grevillea, and here is a coria, which is it's a native ornamental plant. Now, I wanted to show you these because you can start to see a little bit of the system that you might use to classify them, and that Linnaeus thought about, where, for example, if you look at this coria, the leaves are simple leaves. There's only one leaf. The leaf isn't composed of parts. It's only a single one, which is the same with this grevillea here. Each leaf has a petiole, it's attached to the stem, and there's only one. When you look at this one here, each leaf is made out of many parts. The leaf starts here, attached to the stem, and it is split into what I call pinnae, which actually is Latin for feathers, and little pinnules, or tiny feathers. So these compound leaves, these simple leaves in here, and that's one of those split points where I was talking about. If you look at all the wattles, about half of them have simple leaves. They're actually not quite a true leaf, but a simple phylloid, which is like a leaf, and the other half are compound leaves. So there you go. You start with 1,000 wattles, but if you split them by that difference, you've got two groups of about 500 each. Then you start to look at other things. What are the flowers like? Some might have flowers that are little globular round flowers like that. Another main group of wattles has flowers in cylinders. So there you go, you've split each one of your groups of 500 into maybe groups of a couple of hundred each. And you can keep going down characters like that, picking different characters until you end up with a group of 2 or 3 species, and then it's very simple to say, this is that one, that one's that one, that one, that one. That's the crux, the essence of the system. And I wanted to show you, a lot of you may have seen this, we call them honeysuckle in English or Banksia in Latin. I was talking about Sir Joseph Banks, who came in, was the botanist on board Captain Cook's first voyage. So Banksias are named after Banks. And this is one of our most common, a very common bush and even garden plant these days. But how do we go from something like this to an object flat on a sheet like that we can store essentially indefinitely? And that's where we can look at our dryers out here. These are converted old clothes dryers, but in here we just take a plant. I'll get it out and show you. We take a plant and we just put a little bit of pressure on it and give it time and warm air to dry it. And then when they dry, they're nice and flat, which now means if you stack them, they don't break. If you let something like this dry out as it is, you try and store that, you put weight on it, it's going to crumble down to nothing. And that was another one of those sort of crucial discoveries, and that led Linnaeus to have a bunch of specimens that he could handle without breaking and crushing them, and start to compare lots of different plants at the same time. Okay. And that's the essence of a herbarium. We have that collection of hundreds of thousands or millions of plant specimens, and then we can do research at a magnitude that you wouldn't be able to if you only can walk around and see the things that are alive in the bush.
Thanks very much, Miguel. That's been a great introduction to the work that you do here. We might come back here again and look at some other interesting things, because there's some amazing stories in these specimens. Thanks for joining us today on our live Facebook tour. We've got another one next week. Thanks, Veronica, for filming, and we'll see you next time. Take care.